Good morning, everybody. Okay, so let's get started. All right, we pretty much ended up here talking about the modularity of Cree elements, cis-regulatory elements, response elements, and the like. And what I want to do now is just kind of move on to some other kind of funky ways in which you can control expression. And so one of the ways, and this is a really bizarre one, right, is that you can have multiple promoters for the same gene, right? And those promoters can respond differently to regulatory proteins, right? And turn on expression in different cells of different transcripts, actually, and different times, different strength, things like that. So what I actually want to do is, you don't want to look at that. It's an old paper. This is actually the paper that started work on C. elegans by Sidney Brenner, who is essentially the god of C. elegans. Um, what I want to talk about is this, which is a paper that I wrote a little while ago, 7 years ago, actually. How about that? So I worked on a gene called PNC1 for a good while, and it's involved in recycling of NAD, right? So NAD can be consumed. It's not just turned into NADH, but it can be consumed, and that product, the byproduct of that, needs to be turned back into NAD. So anyway, this is what the PNC1 gene looks like, right? So you remember all those pictures that we've shown you of what a eukaryotic gene looks like? Well, this is essentially the same format, right? We have um, 5 different exons, 1A, 1B, 2, 3, and 4. We have a variety of introns, and this is the mutant I was working on, which knocks out function. And there are 3 different promoters, right? We knew of 2 of them when I started, but I found this other one because, basically something didn't make sense. And so I suggested that there was also a promoter here, right? And so you can get 3 different transcripts from this gene, right? Same genetic information, 3 different transcripts, right? They all share these 3 exons, right? So the catalytic activity of this enzyme is in here, right? But you can control where that is expressed and what types of tissues and even the type of protein that's produced, right? And so this first exon here has a signal sequence that directs it to be exported from the cell. So the 1A form, which is this one, this one, and this one, and this one, together, essentially is a secreted protein, right? So this is able to move around the worm. It's not just active in the cells that it's expressed in. Right, so a bunch of this work, not entirely, there's other things going on in this paper, but a bunch of this work was to simply catalog, well, not simply, but thoroughly, there you go, catalog which genes, which cells these 3 different promoters were expressed in. We didn't look at time, in terms of development, because that would have been a huge effort. And we didn't look really in response to stuff. So we actually don't know what controls these promoters, right? We don't know what regulatory elements are present here. And that's actually a very, very hard question to ask. But we do know where they're expressed. And so what we did, or what I did, was I actually made a series of what are called reporter constructs and made a bunch of transgenic worms, right? So these essentially had the 3 different promoters in front of the coding sequence of the two different forms that you can get, this form and this form, right? And that was translationally fused, right? So in frame, remember we were talking about reading frame a while back. In frame with that coding sequence is one for a green fluorescent protein, right? And at the other end, we have the endogenous 3' UTR for that gene, right? So the whole regulatory context, and this is important because things can change a lot if you don't take those precautions. The whole regulatory context is what it should be in the worm itself. It's just that we've stuck a protein from a jellyfish in there that's going to glow green. Anybody got questions about any of that? I know this is kind of, I mean, this is pure research, so this might be something that's kind of step beyond what you may have experienced already. But it's cool though. It's fun to talk about. And as I actually am the one that did it, I'm pretty well equipped to talk about it, which is fun as well. Okay, so what does that actually look like? You don't want to look at that table because it's boring. Lots and lots and lots of work later. We have pictures, right? So these are, let's make it a little bit smaller. Bring up the chat just in case anyone's asking questions. Oh, yes, Jade, that would be totally fine. Yeah, just let's leave it to the end of class. Okay, so essentially these are the different constructs. Let me just double check which is which. 1A, 1B. I wonder why I did it that way around. Anyway, so this is the first promoter, this is the second promoter, this is the third promoter. Right, and so, and this, I think this one is all of them. Let me just double check. D. Yeah, so this is the expression of the whole genomic locus, right, which actually looks different, which is kind of weird as well. So if you blow this up, right, these are the heads of, let me blow it up a little bit more. This is the head of a worm, right? This is the pharynx, that's what sucks in the food. And around here is what's called the nerve ring, right? So that's its rudimentary brain. And it's actually fairly sophisticated in terms of what it can do. Obviously, just isn't a brain like ours, right? And so when you express this one, you see lots of expression in pharyngeal muscles, things like that, salivary ducts. Okay. This is also a white light picture. The reason why it looks shitty is because it's on a confocal microscope, right? And they don't do very good white pictures. This is actually captured using a laser that scans down the screen at a certain focal plane and excites the GFP in that plane. So it's a really cool way of generating very crisp, accurate pictures. Then when you look at the second promoter, now we've got a whole bunch of expression in the neurons, right? So all of these blobs, right? You can actually, if you zoom in a little bit closer, and again, hitting the resolution of the PDF here, you can actually see the lines. Here's one. This is a dendrite, this bright line coming down here. This is going to go to one of these neurons, right? So these are all nerve cells. These are neurons, right? So for some reason, that particular version is highly expressed in the brain of the worm. Don't know why. And then when you look at the third promoter, well, now you don't get any expression in the nerves. Now you get a whole load of expression in the intestine, right? So clearly this protein carries out important functions in very distinct tissues, right? The nerve cells, presumably they're consuming lots of NAD. I don't know for what. And the intestine and some other muscles. Now, finally, and here you can see those dendrites much more clearly. Here's some neurons in the tail. When you look at the secreted form, right? So this is essentially the embryos of a mature adult hermaphrodite, you can actually see here's a 2-cell embryo, right? So that's a 1-cell fertilized embryo. It's already, this one has undergone its first round of cell division, so it's now 2 cells. And these have more cells than you can easily count by eye, right? So these are more developed. Anyway, when you look at this under a different type of microscope, so this is a DIC microscope, which is a more conventional light microscope. Now you can see that there's expression, or presence rather, of that protein in the extracellular space. So this is a secreted protein that's able to move from wherever it's expressed, right, which you can see kind of up here in the other picture, right? So this might be where it's actually expressed, but it travels elsewhere in the worm to do its job, right? So the reason why I'm going into this is because it's just fun to talk about my work, and I'm not ashamed of that. But also it's an exceptionally good example of how multiple promoters can be used to control where that information is turned into protein, right? And that ties directly back to what that protein does in those particular tissues. So anyway, go read that if you want. You'll be one of several people who have. Anyway, I thought that was really cool. I love taking microscope pictures of GFP worms. You might be thinking, well, if this is GFP that glows green, why are these pictures all monochrome? Right, and that's actually because even though it looks green under the microscope, the cameras that we use only capture in black and white. And actually, if you want it to look green, you just do that as like a post-processing effect. And I didn't see the need to do that. I know that it was green. It's green fluorescent protein. So anyway, there you go. So, and there's lots and lots of other ways, you can use this as an organism to control gene expression. The example here that's given is between different life cycle stages, right? So between a adult and a larvae. Pardon me. So essentially, you're going to have a larval promoter that only comes on when that organism needs it in the larval stage, and then there's presumably a different function or use for that protein in the adult stage, and that's controlled by the adult promoter. There's also, do we talk about alternative splicing? Yeah, that's down there. Okay. Now these enhancers, these kind of fall under response elements as well, to a large extent. These really are what determine the cell specificity, right? And essentially the extra bells and whistles of that gene. So where is it expressed? When is it expressed, right? And quite often to what level is it expressed, right? And so these are, these and response elements, they kind of fall into largely the same kind of group. These are sequences of DNA that are recognized by specific proteins, right? Transcriptional activators, transcription factors. And those proteins in turn, their expression is also controlled by other elements, right? So you see there's a multiple layer cake going on here, right? Where a particular gene is controlled by proteins, and the expression of the proteins that control the expression of that gene are controlled by the expression of other proteins, right? And so now you can see how we build up this kind of complex cell and stage specific expression pattern, right? Which allows things to be turned on very specifically where they're supposed to be. And these enhancers don't have to be anywhere near the gene itself, right? They can be all over the place, essentially. And so a really good example of this is two different proteins, albumin. I really should have looked up what albumin does. I know that it's in egg white. So what it does in your liver, I don't actually know. I should look that up. And crystalline, right? And I do know what crystalline does. So crystalline is expressed in your lens cell, right? Or cells. And those are the cells that make up your lens in your eye, right? And so that, or those cells, right, after a certain step of development, need to be clear. Because if they're not, you'll be like wondering, like, oh, it's so dark in here, right? Because there'd essentially be a whole bunch of gump in the way of the light that's entering your eye before it hits your retina. And so that gump, that cellular, material, even the kind of the nucleus and the organelles and proteins and all kinds of stuff needs to get cleared away. And that's what the crystalline protein does. Is it actually forms, it helps clear out cellular stuff, and it replaces it with a crystalline protein, right, which actually helps focus that. Essentially, you've got your lens, light comes in, and then it gets focused onto your retina. That's how you focus your vision, and you kind of stretch it and relax it to be able to change the focal depth, right? So I'm looking at this, and now I look up and see my daughter come downstairs, and the lens changes shape. So anyway, you only need that expressed in lens cells. You don't need it actually in any other cell in your body, just in the lens cells in your lens of your eyes, right? And so this is expressed in your eyes because in those lens cells are other proteins, right? Transcription factors and transcriptional activator proteins, which are only present in those lens cells. And they're only present in those lens cells because prior in development, other genes were turned on in response to other cues, which make that lens cell a lens cell, right? As opposed to retinal cell or corneal cell or, any other stuff that is in your eye. And those transcriptional activator proteins are not present elsewhere, right? Because that lens cell is very specifically a lens cell, right? And the same applies for albumin, right? So you really wouldn't want albumin being produced. It's egg white, right? That's the last thing you want in your eyeballs. Or protein, sorry, in egg white. It's not actually egg white. And so the transcriptional activators for albumin are not expressed in your lens cell. And so as a result, the albumin gene is not expressed in your lens cell. And the opposite is the case for your liver cells. Right, so the specificity of expression, and really in many ways, what makes a liver cell and a lens cell, are the collection of proteins that are present in that cell, right? They're expressed in that cell, right? Because those control the expression of other proteins which are required for that cell to adopt the shape, the function, the whatever it is that that cell does, right? Oh, that bloody cat. Does anybody have any questions? Questions? All right. And here you can also quite nicely see the whole complex that forms, right? So this is RNA polymerase here, and these are all of the, the title binding proteins, the transcription factors, mediators, things like that. Okay, so another quick one is silencers. And so essentially, these enhancers can be all over the place, right? And you don't want those enhancers accidentally activating another gene, right? And so really you need to kind of wall off genes from each other. Oh, hang on, that's insulators. That's down here. Got my timing wrong. And you want to make sure that those enhancer proteins, right, however they bind, don't accidentally activate transcription of another gene, right? Because again, they can be bidirectional, they can be in any orientation. And so those proteins bind to that insulator, and essentially, I mean, typically what they do is they prevent the bending of DNA, right? So they prevent that DNA from bending around so that enhancer can activate an RNA polymerase here, but it can't activate an RNA polymerase over here, for example, right? So that's kind of typically how those insulators work. Now, silencers are essentially regions which negatively regulate transcription. And so there's going to be a kind of a balance essentially between what enhancers are present and what silencers are present. So in this example, this was really focusing on enhancers, but there will also be potentially, depending on the gene and its promoter, potentially there will be silencers that will bind to these regulatory regions and prevent the binding of enhancers, of transcriptional activator proteins. Right, and so this is able to add an extra level of specificity, right? So not only do you have to have activators present, but also absence of repressors as well. And so these can do all kinds of different things. They can compete with binding of the activator proteins, right? They can prevent actually even the binding and assembly of RNA polymerase, right? And so these, essentially in every promoter, you'll have a variety of DNA sequences, right, which are recognized by activators and repressors. And essentially the relative concentration of those different things, their presence and absence, and how much there is present, dictates the level of transcription from that gene. So here's a good example, right? So this is thyroid hormone response element, right? So we have this is a DNA wrapped around histones. We have a bunch of, this is a nuclear hormone receptor, retinoic acid X receptor, I believe, and thyroid hormone response. And so these have to bind and recruit a bunch of other stuff in order to activate transcription, right? So you have to have a whole bunch of different proteins present for this activator to help activate transcription, right? And so this then promotes the metamorphosis of a tadpole into a frog. When you don't have all of those things present, now, and some of them can be present, right? So the RXR and the thyroid hormone response, TR alpha. But if you don't have other stuff present, then you now have a repressive complex, essentially a bunch of co-repressors, and those inactivate expression, right? So this is via acetylation of histones, right? So this is kind of bringing everything that we've talked about together from chromatin formation all the way down to individual proteins, right? So this generates a HDAC, a histone deacetylase complex, right? We've got HDAC3 here that will deacetylate histones in the region of that promoter, and they'll basically shut down its accessibility, even though the DNA is accessible over here for these things to bind. In the presence of other factors, right, which may not necessarily bind DNA directly, now we have a histone acetyltransferase complex that opens up the DNA. You can actually see like the cutesy little arms or tails are now flapping around. Now that DNA is accessible for binding of other things, the basal transcription apparatus, for example. Questions? And so the neat thing is essentially these different combinations of proteins can generate really a very, very large difference in morphology, right? Going from a tadpole, no legs, to a frog with legs, right? Tail, no tail. Well, actually, that starts a bit of a tail. I'm sure we've all seen, frogs in the process of metamorphosis. My kids used to love catching them from the lake. It's just fun to look at them. Okay, so this is all really about control of transcription, right? So as you can see, there are many, many, many ways in which you can do that. All the way from the type of chromatin that you have, right? The marks that you have on histone proteins, acetyl groups or methyl groups, to methylation of DNA or not, to the presence or absence of a range of different proteins which act to control expression of that gene, right? And so you can see even at this level, and we haven't really got any further than that yet. We haven't got beyond transcription. But at this level, you can see the enormous complexity that comes to bear on the control of expression of individual genes. And we have about 20,000 genes, right? And so these bells and whistles and dials and knobs are all present in all of those genes, right? So now you can kind of get some kind of like a little bit of a peer through the looking glass at what's happening on the other side, and it's enormously complex. Okay, does anybody have any questions about transcription so far? Control of transcription, chromatin formation, stuff like that. You already know what exam 4 will be like because it's going to be very much the same as exam 3. Lots of questions on details and what's going on and why. So do take advantage of me being here, able to answer your questions so you can understand this stuff better. Okay, and don't forget as well, most likely we'll have time for at least some catch-up questions, recap questions on Wednesday before our last lab. Okay, so let's get back to this. And so remember we talked about a lot about splicing. Well, obviously its main function is to get rid of introns. But why are introns present anyway? Right, and this is actually a pretty fundamental question which we still don't really know the answer to. But one of the potential answers, at least partially, is if we have introns, we can control which exons are used. Right, so for example, this is part of a gene that's expressed in the liver that is insulin responsive, right? So it responds to levels of insulin. Don't actually know what the protein is. And it's a huge protein, right? So there's at least 8 other exons to the left. I don't know if exon 13 is the last one or not. But either way, when they're all spliced normally, right? So all the introns are spliced out and you have all the exons in order. The resulting protein has a relatively low affinity for insulin, right? That may be because insulin levels are very high. And so you don't want to respond vigorously when there's a lot of insulin around, right? You want to be able to be somewhat insensitive to it. Now, and also don't forget the liver is one of the main organs which takes up glucose out of the blood. Can you remember which the other organ is? What else takes up glucose out of the blood in response to insulin? What else needs glucose? Liver's all about storing glucose in the form of glycogen. What other tissues are very metabolically active and need lots of glucose? And so respond, brain, yeah, that's a good one. Brain uses a huge amount of energy. What else? I'm not using very much of it at the moment for this tissue because I'm sitting my ass on the couch. Pancreas produces insulin. It doesn't necessarily respond to it. Kidneys? Kidneys are pretty much just waste purifying plants. What am I using to do this other than my neurons? Yeah, muscles, right? And so muscles and the brain and liver are the three conventional main sites of glucose uptake from the blood. The brain and the muscles because they use it, the kidney because it stores it, right? So kidney is one of our main stores of glucose. Anyway, so if there's a lot of glucose around, there's a lot of insulin being produced, then you want a relatively insensitive insulin receptor, right? Now, oops, get you back up here. When insulin levels are very low, right, you want to be able to detect low levels of that. And so now that same protein, right, or that same transcript, it's not any different, right? The same transcript is produced from the same gene, right? The information is the same. Now we skip exon 11, right? So we splice that out as if it's an intron. And so now we generate a mature transcript, which will be translated that only has 9, 10, 12, and 13, as well as, 1 to 8, presumably. So we no longer have exon 11. And exon 11 presumably contains part of the protein which decreases its sensitivity for insulin. Because when you splice that out, the resulting protein has a high affinity for insulin, which is very different, right? So the same information is present in the DNA. That does not change. But from the same information, you can generate two different proteins with different characteristics, which is pretty crazy. Right, so that's one way in which you can change. So you can change which promoter is used, right, as I showed you earlier in that example. But you can also change which part of the transcript is used to generate the protein, all from the same information. Okay, so now we're getting into RNA processing, and there's also a really major step, which is RNA stability, right? So if you think about it from this perspective, the control of transcription is all about controlling how much transcript is produced. But also, once that transcript is produced, it will be available for the production of protein as long as it hangs around for. So say you only want this protein produced for a very specific window of time in development, let's say, or response to a very specific condition. After that condition has passed or that time has elapsed, you want to get rid of that transcript because you don't want that protein being produced anymore. And so one of the ways that we do that is by targeted mRNA degradation. And this is actually one of the most important, in my mind at least, discoveries. I mean, there are several that kind of compete for that, right? Discovery, the invention of PCR is one of them. But RNA silencing is actually one of the biggest ones for me. This is a technique that I use or will, once I have approval, use on a regular basis in my lab, right? And this is called RNA silencing, right? And so essentially there are kind of two different kind of entry paths into it, right? And so there are kind of two different outcomes. But essentially what we want to do is we want to either very in a very targeted manner, degrade mRNA, or we want to silence that mRNA so it's not used for transcription, right? That's the kind of overall outcome that we're after. And the way we do that is we produce small non-coding mRNAs, right? So these are mRNA, not coding mRNAs, they're non-coding RNAs there. Let's just type that up. Oops. And so those called, generally speaking, NC RNAs are often involved in mRNA stability and control of translation. And so they're typically very short genes, and they produce a RNA transcript which is not translated. What it typically does is it forms a piece of double-stranded RNA, right, which is rare, right? We don't typically have lots of double-stranded RNA in our cells. We have weird RNA 2D structures, but we don't tend to have much double-stranded RNA. And that, after some processing, will be used like a barcode scanner. And so it will be used to find the mRNA that it matches, right? So that's a really key point, right? So the non-coding RNA has typically a very strong match to its target. And the degree of matching actually changes the outcome to some extent. Does that make sense? Right, so these are essentially like kind of guides. These are little barcodes that will recognize mRNAs in the nucleus and in the cells, in the cytoplasm too. Right, so say you're a worm, right? And you're busy going through your, second larval stage. And time's coming up when you need to molt into your third larval stage. Okay, so how do you trigger that? How do you shut down all the stuff that's involved with just being a second larval stage and trigger the process of molting into the third one? And so one of the triggers is the production of a non-coding RNA, right, which goes around and basically shuts down all of the transcripts involved with anything that doesn't involve molting. And so, and remember I was talking about, the 3' UTR and how that's important, right? And actually that was in one of the example I gave from the work that I did, right? And we talked a lot about, 3' UTR is involved in stability quite typically. And so the region of the mRNA that these barcodes recognize is typically the 3' UTR, right? So in that region of the transcript will be short sequences that are recognized by non-coding RNAs. And so the process is actually really neat, and it kind of depends on what kind of non-coding RNA you produce, right? So you can either produce small interfering RNA or siRNA, or you can produce, which is typically double-stranded, and it's double-stranded because it's part of a very long stem loop, right? So this is the stem of a much larger loop, which has had the end cut off. Or you make a smaller stem loop if you're microRNA. And there are lots and lots and lots of non-coding RNAs. In fact, there are so many of them that I don't know all of the names. There's one called a peewee RNA. I don't know what that stands for. But anyway, lots and lots of them. So first thing that happens is that these double-stranded RNA structures are recognized by an enzyme called DICER. And what dicer does is it processes that RNA further so that we have typically between, I don't know, like 18 and 25 nucleotide long bits of double strand RNA. And the length varies depending on what type of non-coding RNA is. For these ones, it's about 25. For others, it's shorter. It doesn't really matter. Essentially, they're taking this and they're chopping it up into small chunks. Now, those small chunks are recognized by what's called the RNA-induced silencing complex, or RISC. And this contains a protein called argonaute, right, which is very important in what actually happens with these small non-coding RNAs. And so effectively what happens in the RISC complex, it does two, well, maybe three different things. But the first thing that it does is it chews up the non-complementary strand, right? So we've got two strands in this double-stranded little RNA chunk, but only one of those strands will bind to its complementary strand in its target mRNA. So the other one, you want to get rid of it, right? Because you need single-stranded RNA for single-stranded RNA to bind to another piece of single-stranded RNA, right? So you got to get rid of one of those strands. And that's one of the things that the RISC complex, which is kind of funny because it's an RNA silencing complex. Anyway, that's one thing that the RISC complex does, right? So it chews up that unneeded second strand of RNA. Then it uses that and it just goes throughout the cell, trying to find an mRNA that matches. No, 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 no. Going all around any mRNA that it finds. It's kind of holding up that short bit of mRNA and saying, do we match, 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 all over until it finds a match, right? And what happens depends on how good of a match that is. If it's a perfect match, like just kind of clamps on, it's like, yep, dum, 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 all bases match, then argonaute cuts the mRNA, the target mRNA, which you can see down here. And when that mRNA is cut, now we have free ends which are not protected by a 5' cap or 3' poly A tail and binding proteins. Now we have free ends of RNA. And what happens when you have free ends of RNA flapping around? You get proteins that come along, RNases, that will degrade them. So essentially what the RISC complex does in this case, when there's a very good match, is they promote the degradation, the breakdown, or the destruction of that mRNA by cutting it so that there's now substrate or targets for RNases that exist in the cell. And so that mRNA is now degraded. It's no longer present. It's no longer available for translation. That gene, effectively, that the transcript is from, is shut down. Now, the other potential route is if that match isn't perfect. You're right, love. Oh, it's just the phone was going off, love. Oh. If that match isn't perfect, now what the RISC complex does is it kind of goes along and goes, nah, that'll do, right? Not great, but it'll do. Kind of binds to that. And now what it does is it prevents that RNA from bending round, right? And prevents the 3' poly A binding proteins here from being able to activate translation. Right, and so now instead what happens is translation is blocked. And so even though the mRNA is still present, functionally it's dead, right? Because it is not able to be translated by a ribosome. And so also now, even though the mRNA is still present, now is functionally inactive and unable to be translated. So again, you've now shut down expression of that gene. And so I actually use this in my research because I want to be able to shut down expression of particular genes and see what effect that has on the biology of my worms. And so you can do that by using mutagenesis, right? So forward genetics, just by screwing up a bunch of worms and selecting, for mutants. But the issue is that some of those genes are really important. And so if you knock them out, i.e. they don't function anymore because of mutations, the worms don't survive or they don't thrive, let's say. And so what I do is I actually have, well, I don't, but my former boss at Penn State has a library of all, well, I don't think it's all 20,000. I think there are like 18,000 genes in this library. It's a whole bunch of 384 well plates sitting in the freezer. And they contain bacteria which have been engineered to express a short double-stranded piece of mRNA that corresponds to one gene. And so what I can do is I can take that out of the freezer, or even my freezer here, right? I've got a subset of those genes that I've been working on. Take that out, grow that bacteria up in culture, and then use that bacteria to feed my worms. And so the worms consume that bacteria, and somehow via, it's almost like magic, that double-stranded RNA gets into the worm, gets spread throughout the worm, apart from the neurons, which don't take it up for some reason. And that is used to shut down expression of my target gene. And so I'm able to go, oh, those genes are interesting. I've got actually a fairly solid list of ones to work on this summer. I'm going to ask my boss, hey, Wendy, could you get one of your minions to send me da 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 da? And then I get in the post a bunch of plates which have those on, grow them up, knock down expression of those genes, and look to see what happens in my worms. Right, so this is an extraordinarily powerful technique. And actually this, the discovery of this and then the use of it, one guy called Craig Cameron, and who's the other guy? Was it Andy Fire, I think his name was? Two C. elegans researchers won them a Nobel Prize because that's how crazy amazing this is as a technique. Even though it's somewhat mundane now, it's actually kind of incredible to think that you can specifically target and knock down expression of a gene just by feeding certain worms bacteria. Okay, so this isn't just a process which occurs in your cells anyway. It's actually part of the process that recognizes RNA viruses too, and cuts them up because they're obviously not good for you. But also it's a technique that you can use to actually do research, which is also neat. That's kind of why, figuring stuff like this out can be so cool, not just for its own sake, but also for its application in other processes. Anyway, what we're going to do on Monday is go through last few slides of this on post-translational modification, which is pretty neat. And then we'll see what we can get through of the biotechnology and associated stuff before the exam on Friday. So that's it from me. I'm going to stop the share and start this up again. So Jade, I'm just going to end this. Actually, there you go. Here's a question. Jade, do you mind this being our conversation being available? If you do mind, and that's totally fine, I'll end the Zoom now and I'll start it back up so you and I can talk. If not, I'll just keep it going.
No, it doesn't matter to me.
Okay, cool. So everybody else, Jade and I are just going to chat about something to do with the lab report. You can hang around if you want, or if not, you can go off and make yourself a cup of coffee or enjoy the rest of your day. But either way, have a lovely weekend, everybody, and I shall see you soon. Okay, Jade, what's up?
Okay, so I apologize if I sound like an idiot, but Penny doesn't really like explain anything
Oh
when
shoot,
it
did
came to like the lab report.
I got an email from you yesterday, right?
Yeah, it was
Yeah.
late, so I figured he wouldn't respond, but
Yeah,
I was
sorry.
working on it for
I
like a
saw that and I was
good,
like, "Oh, I'm tired. I want to go to
no,
bed."
yeah, I figured, but I was working on it for like a good, like on and off 7 hours yesterday. And I had even met with him in the morning, and he told me that we have to use the gels from the labs that we did. The
No, you don't.
Okay, see, because
Well,
I was doing the
actually,
lab, I
Jade, I've got
just
to be
didn't
very
understand
careful
why.
here. So, yeah, so in my lab, you don't, but I'm not
Okay,
grading your lab report.
yeah,
And so if Mr. Penny requires you to do something, and that's part of how he's grading the lab reports, then you need to do that.
yeah,
Um, and I'll talk with him about that, but I'm a little leery to kind of get into stuff this late in the semester
yeah,
about the lab report because I don't want him to have to change what he's telling his students and sow confusion about what his students have to do to get their grade. And yeah, I mean that's unfortunately, Jada, and I apologize, that's one of the downsides to having different people teach the lab.
yeah.
I
And
can't
like,
teach all of the sections because I just don't have enough time in my schedule to do all of that, which is why we have Mr. Penny.
yeah,
Um, and so if he's said that you need to do that, then unfortunately that's what you need to do.
okay.
Um,
It's just he never throughout the labs, he never really said anything about the lab report. We all
okay,
knew we eventually would have to do it, but he never did like an overview of like, okay, you need to have this in there. This is why. He never did any of that.
right,
And I can understand like we have to put the gels if he said so. I just don't understand what what am I supposed to analyze? Like I don't he never went over like what the gels meant. And that could be on my
right.
fault too, but I don't
So
understand.
so which gels is he talking about? The gels of the PCR products?
He said we had to do all three, the ABC unknown and then the other two as well, because I believe we only have three total. And he told me that I can do the
Okay.
ABC unknown as like a graph on Excel by itself, and then I can combine the other two into a graph.
Oh,
But
I'm
he just
sorry,
doesn't
Jade.
tell me why. And it's like I don't want to put it and then I'm explaining something.
Yeah, I mean, I
Yeah.
wouldn't know what you would use that for because that's a completely different experiment. That has nothing to do with this.
Yeah.
Um, I did send you the linkage report kind of Word document, right? I think.
Yeah, I've been going through all of them as I
Right.
was writing it just to try
Okay.
to make it make sense.
So the unknown ones, that's just a completely different experiment. I'll have to talk with him about that because that's I wouldn't even myself know what to say about that.
Yeah.
The other ones, I mean, really, at the end of the day, you put them in there and you say, "This is what we found. We had DNA. That was good." You know,
Yeah, I
we
figured
had
that's
a
what I would just do, just do
yeah.
something kind of general. Because he
Well, I
told me
mean,
he was
that's
going
all
to
you
be
can do.
following the rubric that you provided, that he was just going to be following that.
Okay. Well, I would put those second two gels in there anyway. It shouldn't take you an awful lot of work. Hopefully, it's just cut and paste. Then it's, figure one is DNA gel, we had DNA. Figure two is
Yeah.
PCR gel, we had a PCR product. And that's really all that you can say about them. I mean, it's not a bad thing to do. I mean, I don't require of my students because it doesn't really have anything to do with the linkage stuff, right? It's a completely different
Mhm.
experiment. I don't know. I'll have a word with Mr. Penny and see if can strain them out on that. It's a little bit of a challenge doing it this late in the day because, again, I don't want for him to send out conflicting messages to students that then results in
Yeah.
confusion and creates other problems.
Mhm.
But
As far as I
yeah.
know, because for the other girls in my lab, we kind of made like our own group chat. So
Mhm.
that way we can like help each other out.
Oh, that's good.
None of them have no idea what's going on either. I think I'm the first one to even started on
Right.
the lab report.
Did Mr. Penny upload those files that I sent you? Because I sent them to him as
Yeah,
well.
he did. He never said
Okay.
anything about it, but he
Okay.
uploaded them.
Okay. I'll send him an email as soon as we finish here before I forget.
Okay.
Um, but at the end of the day, you need to follow what your professor's telling you, because
Mhm.
I'm unfortunately not your professor for lab.
Yeah.
Um, but I will try and make sure that what your professor's telling you is what he should be telling you as best as
Mhm.
I can. Um, yeah, and I'm sorry that I haven't caught this, until now,
No, yeah, it's
um,
fine. It's
because
just
I
I
don't
want to
know
finish
exactly what
it.
to do. Yeah, I know, and I really don't like the idea of one set of students being assessed in one way and a different set being assessed in a different way, even though they're doing the same thing and supposedly getting the same grade.
Yeah.
So that's an issue. But I guess this is the first time I've actually worked with Mr. Penny to teach this stuff because I was supposed to be working with him last semester in the fall,
Mhm.
but I was sent on an unpaid leave of absence because the university screwed up my immigration stuff.
Mm.
And so I didn't work for the full semester. And he taught all of genetics lecture and lab for the full semester. So I didn't actually have an opportunity to catch any of that stuff then because I
Yeah.
wasn't working. And so this is the first one. And so there's been a series of kind of like going on, and it's just part of those teething troubles. Not
Mhm.
much of a consolation for you, obviously, because hopefully you're going to pass and you're not going to take this again.
Yeah, hopefully.
Um, but it's just part of that process of essentially training someone else to teach the lab.
Mhm.
Yeah, so I appreciate being made aware of this stuff because otherwise I'm essentially clueless. Um, but I'll see what I can do and send him an email or maybe Zoom with him and see if we can clear this up.
Okay.
Yeah, but
But I
in the
figured
mean,
I would just try all my bases.
yeah, and it's good to know this stuff because otherwise I don't have any insight into how the lab is being taught. And I do want to make sure that it's an equivalent experience for all of my students, not just the ones that I happen to teach lab for. Um, but yeah, I'll get onto that and see if I can sort that out because I think it's just most likely a misunderstanding on Mr. Penny's part about what he's supposed to be doing.
Yeah, that's what I figured because like I asked him, I sent him the same email and he just told me this morning, he just told me that we're using the data from the gels and we're not using our fly counts. That's all he said. And I know we weren't using the fly counts because we were provided those counts because the flies died, but that's
Right.
literally all he told me. And I was like, that doesn't explain anything, but thank you.
Okay. Yeah, well, I'll get onto him and see if I can give him some better information to work off of
Oh
because
yeah.
I don't know how he did this last semester. I've absolutely no
Yeah.
idea. Cool, but thank you anyway, Jade, and I'll see if we can figure that out.
Okay. Thank you.
No worries.
Mm.
On JM for my lab section, the lab report final draft is due on the 7th of May. Now, the rough draft, if you want feedback on that, that can be any time between now and I don't know, like 3 days before the deadline. Because if you get me your rough draft on the 7th of May, I'm not going to be able to get it back to you in time for you to be able to use that to change it and resubmit your final draft. So if you want feedback on your lab report, send it to me a few days ahead of the deadline at least, and I'll be able to get that back to you. But the actual final draft is due on the 7th of May. Awesome. Thanks, everybody. I shall catch you next week. Have a great weekend.